Hi, my name is Josh, and today we'll be talking about the KRAS protein and the G12C mutation that occurs on the KRAS protein. The KRAS protein is a relay molecule that is responsible for relaying signals from outside of the cell to the cell's nucleus in order to instruct cell growth, proliferation, and death. It is located in the cellular membrane and cytoplasm of the cell. It is found in any cell that undergoes cell growth, proliferation, and death, which is epithelial cells, connective tissue cells, immune cells, and nerve cells. It is found exclusively in animal cells. So typically, the KRAS protein is responsible for acting as a switch while converting between two molecules GTP and GTP. When an extracellular signal binds to a cell that facilitates the exchange of GTP for GTP, this is when KRAS enters an active state. The active KRAS is bound to GTP, which interacts with the downstream effectors that initiates a cascade of protein activation. This leads to the activation of the RAS MAPK pathway, which signals for the activation of MAPK to initiate phosphorylation and regulation of various transcription factors. This leads to the expression of cell growth, proliferation, and differentiation. When the process is complete, the KRAS hydrolyzes from the GTP, causing the change back to GTP in order to instruct an inactive state. So for my KRAS molecule, I created out of pipe cleaners this globular shape. So the KRAS protein is typically a globular molecule, and I use the pipe cleaners to just demonstrate how fluid a globular protein is. So the KRAS protein is a GTPase consisting of a G domain that binds to guanidine nucleotides. It also contains a hypervariable region, which we can see here, that facilitates its membrane association. And we have 5 alpha helixes and 6 beta sheets present on this molecule to represent the protein. The subunits of the KRAS is typically a monomer, so its primary shape is just a linear sequence. Its secondary is these alpha helixes and beta sheets we see, and then the tertiary, the third structure, is just this globular molecule, and it doesn't have a fourth structure. So for the mutation, I put this X here on the hypervariable region where this mutation occurs on codon 12. So the G12C mutation is a missense mutation that occurs on codon 12 that changes glycine to cytosine. This mutation causes the regulated protein to switch to a constant state, being active, where the KRAS protein cannot hydrolyze from GTP back to GDP due to impaired ability of the GTPase, which causes the protein to be in a locked or activative state where it is constantly sending signals for cell growth and proliferation. The G12C mutation has a profound effect on the protein's core function as a molecular switch in cell signaling that disrupts GTPase activity that consistently activates the cell and alters its interactions, which can affect the cell by leading to uncontrolled cell growth, proliferation, disturbed differentiation, cells being able to survive under conditions that would not normally trigger cell death, and effects on metabolism. When looking at the effects the mutation can have on the organism, it can lead to the formation of tumors, which can be an early driver for the development of cancers, which is why we see this mutation often lead to cancerous diseases being present, such as we see colorectal cancer, lung cancer, and pancreatic cancer being some of the major diseases that are present from this mutation. So the normal mutation I used to represent here is where glycine is present, and then this one where the mutated version where cytosine is present, which all occur on this hypervariable region at the X. And for 3 possible limitations of the variable is that it's just a simplified representation of the overall 3D structure. It doesn't capture how dynamic the movements and interactions of the protein are. And that a second one is that it's just a static model, so we're not seeing it actually interact or how this mutation actually occurs within the cell. And then the third one, we can't see the membrane effects of the 3D model, how it interacts with the protein lipid bilayer. That is very crucial to the overall protein's function. But I believe that is it. Thank you for coming and watching me talk about the KRAS protein.